Okay, we're back for video number 2, and we want to continue talking about cellular respiration. And one of the things that we want to remember is that if you remember the formula from some of the previous slides, we had glucose plus oxygen, and then we said that it would yield carbon dioxide and water. So we're saying that this is a catabolic reaction that requires oxygen. What we'll find out, and maybe what we mentioned before, is that oxygen is going to be very important because it's going to pick up those energy spent electrons from the electron transport system. So it requires oxygen in order for us to do or complete the breakdown of glucose. So this reaction often involves the complete breakdown of glucose into carbon dioxide and water. Now, we know that glucose is a high energy molecule because of all those bonds that are in glucose. If you remember, we said catabolic reactions, they release energy. So we know that glucose is a high energy molecule, and what we're going to find out is that as we're breaking these bonds and these electrons are being lost, so we know that anytime electrons are being lost, that's called oxidation. We learned that back in the chapter dealing with energy. And so and when we pump these hydrogen ions around across membranes, we know that when they flow back through the ATP synthase complex, that's where we're going to be building that ton and tons of ATP. So mitochondria use the energy that is actually being released from that slow breakdown of glucose. So if we look at the actual formula, glucose plus oxygen yields carbon dioxide and water. It looks very simple, but there's a lot of intermediate steps in the breakdown of glucose. Now, the two coenzymes that are going to be available to us are nicotinamide adenine dinucleotide and flavin-adenine dinucleotide. So these are the coenzymes. These are the guys that are going to be picking up the electrons as they're being lost. These are the guys that are going to be picking up the hydrogen ions as they're being lost. I refer to them a lot of times as the cab drivers because they are picking up their fare, which are the electrons and the hydrogen ions. So again, these are our oxidation reduction coenzymes. Now, and the what you see are the oxidized versions of the coenzymes. When they pick up the hydrogen ions and the electrons, they become the reduced versions of the coenzymes. So for example, NAD plus will become NADH, FAD will become FADH2 once they pick up their fares. All right, so this is again what we could call cellular respiration in a nutshell. We begin with glycolysis, and as you can see, glycolysis is taking place outside of the mitochondria. It's taking place outside of the mitochondria. So here we are. We're outside. So here's the mitochondria. This guy over here. I don't want to, yeah. And so it says this has to be the cytoplasm of the cell because this is where glycolysis is taking place. It's taking place in the cytoplasm of the cell. It's taking me a long time to write this. Sorry. And it says during glycolysis, it says what we're going to see is that glucose will eventually be broken down completely into pyruvate. So remember, if you remember the name for glycolysis, it is a sugar splitting reaction that breaks glucose into 2 3-carbon molecules, okay? And so at the end, we will actually end up with not 1 pyruvate, but 2 pyruvate. One of the other things that this shows us is that we're actually going to have some ATP generated during this reaction. And the other thing, it also shows us that we have a coenzyme. Actually, NAD is going in. Remember, NAD plus is going in. It's picking up hydrogen ions and electrons. Look, it's showing electrons. Also hydrogen ions, and it becomes, so it says he gets reduced to who? NADH, because it's picking up hydrogen ions and electrons. Move over to the transition reaction. It says we're moving into the matrix of the mitochondria. So the transition reaction or preparation reaction moves us into the matrix of the mitochondria. Here again, we're breaking down pyruvate, and it says we're going to pick up some hydrogens and electrons. So again, NAD plus will be reduced to NADH. And notice where all this is going. It's going, if you follow the arrows, all arrows lead to who? The electron transport system. It says, did we make any energy? Did we? None. There's zero energy created, no ATP. But then the citric acid cycle also taking place in the matrix of the mitochondria. It says here, it says we're going to store some electrons and hydrogen ions on our coenzymes, our cab drivers. And so we're going to actually create NADH and FADH2. Again, all going where? To the electron transport system. And it says during the citric acid cycle or Krebs cycle, because remember we said it had two names. It says we're actually going to create some energy here. So we're going to get 2 ATP here as well. But then look at the electron transport system. It says that's where we're going to build the majority of the ATP. It says here we're going to produce 32 to 34 ATP during the electron transport system. And if we add that up, it says we're going to get 36 to 38 total, because here's 2 and here's 2. So that's 36 to 38 molecules of ATP. All right, so let's look at glycolysis. Remember we said it's a sugar splitting reaction. It says for these, one of the things that if you remember from one of the previous videos, I said things that we want to remember. What is the starting substrate? Let me grab a pen over here. So what is the substrate? Okay, so this would be the substrate. Or reactant. We could use that word. So this is just the beginning substrate. There's a lot of guys in between. So if you remember a long time ago in chapter 6, we talked about metabolic pathways. This is a metabolic pathway that involves lots of enzymes in order to get to the final products. And then so we said pyruvate is the product for this reaction. Remember we said it's broken down into 4 reaction groups. So this is the first of the reaction groups. Okay, and then the other question that I asked, did you create an intermediate? So we're going to get there in a minute. All right, so during glycolysis, it says that glucose is broken down in where? The cytoplasm and the final product are 2 molecules of pyruvate. Because remember, we said this is a sugar splitting reaction. So we split glucose into 2 3-carbon molecules. Okay? Now, it says in order to activate the splitting, it says we have to put in 2 ATP. And 2 ATP are used to activate the splitting of glucose, which is, we know, a 6-carbon molecule, into 2 3-carbon molecules that we're going to call pGal. Now, here we go. P-gal then is the intermediate. Okay, so P-gal is the intermediate. So I'll write that word over here. Intermediate. It is the intermediate. That's created that will eventually, these will become the 2-pyruvate. So we have 2 P-gals. So this is our intermediate. Okay. And then it says 4-AT will be gained once we oxidize P-gal down to pyruvate. Now, it says how we gaining these ATP. We gain these ATPs through a process that's called substrate level phosphorylation, where we go back into the substrate and we cleave off the phosphate groups. Those phosphate groups and putting them onto ADP allows us to create ATP. So what we're going to find out, so the net, if you remember from the previous slide, it said that 2 ATP were created. Well, I guess maxed for, or the net for this reaction. And the reason why we say net is because it says in order to activate the splitting of glucose, it said we had to use 2 ATP. So we know that that would be, if we used the 2 AP, we know that that's like a minus. I'm still not grabbing a pen here. So that's a minus 2 ATP. But then it says we gain 4 ATP when we oxidize P-gal down to pyruvate. So we know that that's a plus sign. So a -2 and a +4 gives us a net 2 ATP for the whole reaction. Okay, so one of the things that's here, it says substrate level phosphorylation, which is what I mentioned before. It says when we take a phosphate group from the substrate and we add it to, so that's substrate level phosphorylation. So when an enzyme comes in, cleaves off that phosphate group, adds it to adenosine diphosphate, that's how we form ATP. And if you want, I can come back later and show you that process. It won't be required on the test to show me the process, but to understand the process, yes. All right, so here's the energy investment phase. So remember we said we had to use 2 ATP. We had to use 2 ATP. So in other words, we phosphorylated glucose. And since we had an input of 2 ATP. But then it says when we the energy payoff phase, so now we split glucose into 2 3-carbon molecules that we call P-gal. Then it says we have an energy payoff phase where we go in and take those phosphate groups off of the substrate. Well, the enzyme will actually do it. And it says this is an energy payoff phase. So it says we're going to gain 4 ATP during this process. So again, it says then our net would be, because we know that that's a minus 2 ATP, 4 ATP. So this is a net for the reaction R2 ATP. So that would be our net. And then it says the 2-pyruvate will go into the next reaction, which we know is the preparation reaction. The other thing that it shows is that we stored some electrons and hydrogen ions on our nucleotide coenzyme, which is NAD+, And we have the reduced versions because they picked up the hydrogen ions and electrons, and we have two NADHs created. So it says the inputs, we put in glucose as our reactant, we got out pyruvate. We stored some energy on our coenzymes, our cab drivers. They picked up hydrogen ions and electrons, and we ended up with two NADHs at the end of this reaction. And then it says, as far as ATP, the total netted for this reaction would be 2 ATP. Okay, so I'll see you again in the next video.